Hello, welcome to activity number 9, evidence-based medicine. This is the article that we are going to be using. And we start with a clinical question, which is, which are the methods that provide the highest level of evidence in terms of effectiveness and application of the use of certain agents in the treatment of osteoporosis? First, we have that a panel of experts in osteoporosis and evidence-based medicine applied rigorous and validated scientific standards to produce a systematic review and meta-analysis of randomized controlled trials of anti-resorptive agents used to treat osteoporosis. And the results of meta-analysis are currently the main source of summary evidence for the efficacy of treatment for a specific condition. And then we have, of course, our hierarchy of clinical evidence, that we know that large randomized controlled trials and meta-analysis of said trials are at the top of the hierarchy of clinical evidence, and medical decisions should be based on the results of such studies. And well, of course, we have that the meta-analysis combines the results of multiple trials, some of which may have a limited ability to demonstrate a treatment effect due to their small size. And that is precisely why large trials are better able to identify treatment effects rather than smaller trials and provide more precise estimates of magnitude of said events.
Based on the foundation of evidence-based medicine, searching the best evidence from literature, in this article, the osteoporosis methodological group investigators define and strictly adhere to detailed specific methods for the systematic review to help ensure that reviews will be comprehensive with reductions of bias. Only randomized controlled trials, the highest level of evidence, were included, and the methodological quality of each trial was determined using formal objective criteria. Also, based on this foundation of searching the best evidence from literature, in this article, two general mathematical models with different underlying principles are used in meta-analysis: fixed effects and random effects. Bone mineral density, vertebral fractures, and non-vertebral fractures were the outcomes of interest in the osteoporosis methodological group. Meta-analysis and separate analysis were conducted for each of these outcomes for each of the agents. The validity of a systematic review may be compromised because trial results may be published more than once. To identify and to limit this effect known as publication bias as much as possible, and based on the foundation of evidence-based medicine, critically appraising the evidence to assess its validity, in this article, data from unpublished studies are included in the meta-analysis, duplicate publications are identified and omitted, and statistical tests are performed to detect such bias.
Meta-analysis of levodopa, residronate, etidronate, relaxofene, and vitamin D found a significant reductions in vertebral fractures, risk ranging from 36% to 48%. Only alendronate and residronate significantly reduction the risk of non-vertebral fracture. Residronate was the only other anti-receive agent to show a significant reduction in the risk of non-vertebral fractures. The rest gain in bone densitometry of the lumbar spine were observed with alendronate, residronate, and hormonal therapy.
Talking about the discussion and applying the efficacy evaluation of this application of evidence on a patient in areas of improvement, we have that discontinuation due to adverse events was a decision, and the adverse events in the improvement area, and the tolerability of all the agents examined was good, and only vitamin D and barely rexofene had discontinuation rates that were significantly different from placebo, and that's the evidence of on the patients. Also, because of the risk associated with long-term use of hormone therapy, that's the evidence, and the improvement area, is it no longer recommended solely for prevention of treatment or osteoporosis, and that's the decision on the patients. Even that the hormone treatment significantly reduces risks of both vertebral and non-vertebral fractures.